Hello everyone, my name is Gerardo and I love viruses so much. I keep talking about it and it's scary for some people, especially for my grandma. My grandma, every time she calls me, she tells me that she's praying for me. And I understand that there's a stigma about viruses being scary, but I want to begin with the history of viruses and also the history of my passion. So everything began in 9th grade when I was in biology class and the teacher was telling us about different scientists and how they changed the world and shaped it. But I was obsessed with this guy who is Edward Jenner and his whole experiment, mainly because he was so unethical, but also it changes so much lives and it helped to eradicate the smallpox. So the smallpox is a deadly virus that causes severe skin lesions and it has been around us for a long, long time. There's some evidence of mummies with smallpox lesions and there are some cultures that even have deities they had to pray so they didn't get smallpox. In the 1700s in UK and Europe, 30% of the people that got infected died. That meant that 100,000 people every year died. And those that survived, 1/3 went blind because they had lesions in the eye. That was the context that in 1749, Edward Jenner was born here in England, in Berkeley. So he was a physician in different hospitals, and there was this rumor that milkmaids didn't get smallpox because they were infected by a similar disease that was cowpox. So he wanted to put this into practice, and his solution was to basically get an 8-year-old boy, who was the son of the gardener called James Phipps, and after scrapping some lesions from the hand of a milkmaid, he will just scrap it in both of the arms of the kid until he gets sick. And he got sick of cowpox, but he was able to recover. And once he was recovered, he was just stabbed with smallpox. But he survived. So then he was stabbed again, but he survived. So then he realized that the rumor was true. If you get cowpox, you won't get smallpox. He did this 23 times again. Everyone survived. So 2 years later, in 1798, he published his discoveries and created the first vaccine that comes from Latin for cow, vacca. Without knowing, he discovered that if you get infected by one disease, you can create a memory that can save you to protect you from another disease as long as they're similar. He didn't know, but these two viruses were from the same family, poxviridae. And as I mentioned, in 1980, the World Health Organization declared smallpox completely eradicated, the first virus and the only virus so far that we've been able to eradicate. But he didn't know it was viruses, he just knew it was diseases. It took almost 100 years to discover that they were viruses. So this guy, Dmitry Ivanovsky, was a botanist from Russia, and he discovered that after putting some leaves through a filter designed to trap bacteria, some materials went through it. So he discovered that there was a smaller agent, and it was the tobacco mosaic virus, which is a virus that just infects plants like this. But then, as now we know, viruses are risky and they're scary, and working with that, it's really dangerous. That's why the levels of biosafety containment had to be created. It was proposed in the 1940s, but it wasn't until 1984 that there was an actual manual for it, called the Biosafety and Microbiological and Biomedical Laboratories, published by the CDC and the NIH. Keeping the idea on the metaphor of a family, I want to propose the metaphor of a house in which you have different levels, being the first level the safer one and the last one the hardest. Because not all of the members of the same family can reach really high. For example, a baby or a grandma, they can't climb really fast to get to the last. But the rebellious teenager with a lot of adrenaline and hormones can run really fast and be really problematic. So in the first level, you have the non-containment. These are viruses that only infect animals and plants. For example, the tobacco mosaic virus. You go one level up, you get to the level 2, which have moderate disease. You can get sick, but it's not likely that you're going to die. For example, influenza. One more, high containment. These are a serious disease, and that's where coronavirus is right now. And then you get to the last one, which is life-threatening, max containment, such as Ebola. Every time you climb up the stairs, you need more preparation, you need more resources, but also the investigation is more risky, so you need more protection. When you're in the first level, it's suggested that you use gloves, and a lot of people don't do that, especially if it's plants. They'll just touch the infected plants like nothing happened. When you get to level 2, it's obligatory to use PPE. Well, that's me in a lab here in uni. You need gloves, you need a lab coat, and you need something to protect your eyes, like eye goggles or a face splash. And if you're working with pathogens that travel through air, you also need this, that it's a biosafety cabinet, which is just a device that makes sure that what's inside remains inside and what's outside keeps it outside. When you go up one level to level 3, the biosafety cabinet's still there, but there's more protocols and more things that are required. For example, the labs have restricted access, which means that you have to follow some tests to be able to go there. So not a lot of researchers can do that. And actually, even if you have a PhD, that doesn't guarantee that you can work on a level 3. Also, you need specific equipment for that, such as a suit that will only be used there, and also different layers of protection, for example, gloves. Also, the whole lab has to have a better system of flow air and filtration. So as we can see, just going up one level requires a lot of investment. And finally, you have the level 4, in which you need this type of suits, but there's only 50 in the world because the investment is so big. Now that we know that, we have a huge problem. How can we make research more safe, and how can we make it more accessible for people? So the first case I want to talk about is this one, when you have feline calicivirus, FCV, which is a level 1, and it's a virus that affects cats with some lesions in the mouth. And it can be used as a surrogate, which is just an alternative for the human norovirus, or the vomiting bug, which is a virus that basically causes gastroenteritis. And it's a level 2, because you can get sick by it, but it's not likely you're going to die from it. So in 1998, Cannon and colleagues studied the effectiveness of disinfectants against norovirus, but they realized that norovirus has a problem in growing labs. They're really non-stable, and try to culture it, it's so hard. So they decided to use FCB, and it was successful. So 5 years later, in 2003, the United States Environmental Protection Agency recognized it as a suitable model. Today, it's still used to evaluate hand sanitizers, surface disinfectants, and antiviral drugs. How? Because they're part of the same family, the calicivirus, and they have a trait in common, which is the caspid structure, which is this right here. All you need to know is that if you're able to break the green part of the cat virus, you can assume that you can break the green part of the human virus, because they are the common trait. In a similar way that when you go to the hospital to get your eyes checked, they're going to ask if your parents have it, because if your mom and your dad has it, it's really likely that you're also going to have eye problems. Another example are coronaviruses. They work as alternatives or surrogates for each other. And coronavirus is a family that we know because of COVID-19, but they have been around us for a long time. And there has been a lot of epidemics throughout the world. For example, the 2003 SARS-CoV-1 epidemic and the 2012 MERS epidemic. All of the viruses have the similar structure. They're called corona because it's it's Latin for crown because of these structures right here. But they also share another common trait, which is the genomes. They have similar structures such as this or this. That's when, in 2024, for my honors project, 3 days a week, for 10 weeks, I had to go to this building, the Center for Viral Research, to try to create a reverse genetic system from NL63, which is a level 2 virus. That only affects kids, but it's mildly. So the reverse genetic system is a tool to manipulate a virus genome in a controlled way. So you can study different stuff like the genes, but you can also finally produce vaccines with it. We chose NL63 because it uses the same receptor as COVID-19. So that's, again, a common trait. After a lot of science, these are my results. They were able to modify the OC43 genome before I arrived and put a protein marker, which is a protein that under some circumstances will be fluorescent, showing some green color. If you see a green dot, that's a cell that's infected. If you don't see it, it's not infected. Something like that can be done with my tool, but we didn't have enough time. So we had to recur to another common trait, which is the cytopathic effect. Basically, coronaviruses, when they kill, they leave a gap, like a circle, and you can assume that that was a coronavirus infection because of that, which is this pattern here, if you can see. After comparing, you realize that my tool looks a lot like the one they already had, and it didn't look at all like the samples that were not infected. So at the end of my honors project, we had an NL63 tool that worked. We were able to determine that NL63 has a similar behavior to OC43, and now we have a new entry to the coronavirus tool library, which means that we're prepared for the next pandemic better. But we have to remember that NL63 and COVID-19 use the same entry, but they don't use the same entry at OC43. So that's when you have to recruit to other common traits. So what have we learned? Since Jenner experiment, in 1798, we were able to first discover virus, and then we learned that his project was so immoral and so unlawful, so we had to create the biosafety levels. We had more understanding that stabbing kids maybe is not the best idea to do science, but that helped us to understand common traits. And using common traits, we were able to use surrogates to first minimize risk and also maximize research opportunities. I never thought that as a bachelor student from Colombia, I would be able to put a little grain to the knowledge of what was COVID-19. Since Jenner's experiment in 1798, I was able to do my project, and today, proudly and with a lot of honor, I was able to give this presentation. Thank you.